Just to continue our Sturgeon Genome part, it's my pleasure to invite Manfred Šartlь to continue this exciting talk of collaboration, but taking a view from German side, from the European side. And it's also, I tried to open this question in the first day, because Manfred is one of the most famous scientists in the field of comparative genomics, but in a very difficult field, because fish genomics is quite tough. So this choice of working with a really hard topic is quite interesting for me to know how it's going. Okay, please, Manfred.
Yeah, thank you very much for the kind introduction, and I also thank the organizers for bringing me to Sochi. It's a great experience. I enjoyed this meeting a lot. I learned a lot, and I made a lot of new people that I've met, and it's just a wonderful experience. So I was asked to give you a short overview about myself. As you see, I've studied biology in the beginning, and at that time becoming a scientist was kind of an unsecure plan, so I also added some studies to become a teacher, which gave me a good education in chemistry. I've been for a long time at the same place, which usually is not a good idea. But for me, it was the only possibility to follow up a research, a line, an area which was only at the University of Gießen by my PhD mentor. Then I moved for a postdoc. I worked with Bob Gallo. This was before he found the human immune deficiency virus. He has studied cancer a lot, so I did my postdoc there, then returned again to Gießen to follow up on my pet research. But then, of course, it was time to move away. Then I became a junior group leader at the Max Planck Institute in Munich, where I also did my habilitation. By the way, my habilitation mentor was also Charlie Davids, the same person who was a mentor of Thomas Bosch that we heard before. And after the time in Munich, I became full professor for biochemistry at the University of Würzburg, where I stayed all the time. I have to say, and this is maybe also an advice, as you can see from my CV, everything was in Gießen, and there was a plan to bring me back to the same place where I've grown up, which then, due to some unforeseeable incident, didn't work. And I can tell you this was the best thing that happened to me in my life. Don't go back to the place where you've grown up. It's a better idea to go somewhere else. So from Würzburg, I had several sabbaticals and visited places, had offers which I rejected. And then in Germany, we have a mandatory retirement age, which on the one hand is good because you have to make space for the young people, for the talented one. You should not sit on your position all the time. But we have the situation of what we call a professor emeritus in Germany. So as long as you have grant money from the research council, you are allowed. And it's a very pleasant situation because you don't have any obligations except for doing science. So you can concentrate on the thing that you are loving most. And I've also have a second position, which is in Texas, where I also have a research position, which means that I'm moving between two worlds, and I'm frequently at these airports when, hopefully, COVID allows us to move again more freely. So, my main research, and this is why I was all the time in Gießen at this university, was to study a peculiar animal system. This was already detected in the 20s of the last century, that if you cross certain fish which are apparently healthy, that the offspring will develop kind of a skin cancer, a melanoma, which is absolutely comparable to human melanoma. So this has been a great model for me to learn about the molecular biology of cancer formation and use this knowledge from a laboratory animal and use it for clinical applications. There is also, but this is a genomics meeting, so there is some genomics behind that, but the more interesting part would be biochemistry, signal transduction, and so on. So this is not the topic of today. I will talk about my second interest, which is comparative genomics, and now I will give you a few more information about the project that we've been doing with Vladimir over the last years. We have been interested in sequencing the first Sturgeon genome, which, as Vladimir explained, is not an easy task because it's a polyploid genome. And this, for a long time, was impossible because the technology was not available. But with the so-called long-read sequence technology, we could approach it. We have an assembly, so the genome size of we took the sterlet, which is one of the smaller species. So the genome of this fish is a little more than half of humans. Also, it has a double chromosome set. The sequencing technology allowed us to assemble the genome on a chromosome scale. All the 60 chromosomes are assembled one by one. And also for those who are the experts, they will see that the Busco report, which tells us the completeness of the genome, we have a 96% Busco, which is almost as best as one can do. So when we did the genome assembly to put a genome together in chromosomal parts, which is our ultimate goal, we let's say we misuse something that we have heard yesterday, namely information from the 3D genome. You've heard that there are special methods for linking those regions in the chromatin which interact. And the people who are studying the three-dimensional genome, they are more interested in the long-distance interactions. But these data also give us the short-distance interactions, meaning those pieces of the genome which are together on a small scale. And this is what we can extract from these data set and then put the small sequence pieces together which are in one chromosome. And this is what you get from a regular fish genome. So this is a fish from Mexico, and each one of these squares is one of the chromosomes. And this is the picture that you would expect. This is a regular diploid species, and this is what we got from the sterlet. And you see it's not as nice, so this chromosome 1 in the left corner looks nice, then 2 is also okay, then chromosome 3, we get all this background. And this needs a lot of manual curation. So the bioinformatician has to sit down and check all these dots and see, are these short distance or long distance interactions. And the problem with a sterlet is that every chromosome is theoretically there in twice, not just in one like in a diploid. And with a lot of this manual curation, we were finally able to assemble all the 60 chromosomes, but you see there are still areas which we cannot resolve, but I'm sure these might be of interest for people who are studying the 3D genome because these are the long-distance interactions. In fact, you can see that the N50 value, meaning the number of scaffolds which have 50% are larger than 50 megabases or so. This tells us that we have a quite reasonably good assembly. What you see here is a comparison on the left. On your left side, these are the chromosomes of the gar. The spotted gar is a fish which did not underwent the fish-specific genome duplication. So it's certainly our outgroup. It's the most simple fish genome. And then on the right side, you see the chromosomes of the sterlet. And these lines which connect homologous regions, we see that almost that all the chromosomes of the gar are connected to 2 chromosomes of the sterlet. And this is the first indication that the sterlet indeed has a full duplication of the chromosome. We call this the double conserved synteny. When we then look into the sterlet, which is on the other side, and just look which chromosomes match together, where do we find homologous genes? You can see that chromosome 1 and chromosome 2 are almost identical in gene content over the whole length. And this is true for several of the large chromosomes. The medium-sized chromosomes obviously underwent some rearrangements, translocations, and inversions. And there are a few chromosomes in the smaller range where you see the red arrows, which do not have a counterpart at all. This will be interesting for us in a minute. What I've shown you so far is what usually genome watchers and genome sequencers and de novo assemblers have, namely the result of what the computer tells you. So it's just the bioinformatic result, but of course we are not sure how real is this. Is there a reality behind it? And luckily for the Sturgeon, we have Vladimir, who is the world specialist on microdissection of chromosome, and as he has mentioned, from the sterlet they have these microdissected pieces of the chromosomes, and these microdissected pieces were sequenced, so we could use the sequence from these microdissected chromosomes and then ask where in our assembly do they map. And this is just one example. So we got from the sequences from Vladimir from one arm of chromosome 1 that he had isolated. And when we map this sequence against our whole assembly, you see that it's half of chromosome 1 and the corresponding part of chromosome 2, which are identified by this sequence. So and we've done this for all the chromosomes, so we are pretty sure that what the computer tells us is also the reflection of the cytological situation. We have already heard from Vladimir that these whole genome duplications have occurred very frequently, in particular in fish. There is here this whole genome duplication which happened long ago, which all vertebrates are sharing, but then there are several polyploidization events which are specific for fish. There is what we call the 3R, which is common to all the modern fishes, and then even within these there are additional polyploidizations, for instance in the cyprinids and also in the salmonids, and so what was suspected from chromosome work, also the sturgeons underwent specific genome duplication. So the question was, when did this happen in the evolutionary past? And I'm not going into details of this analysis, but when we compared the genomes of different sturgeons and calculated the evolutionary age and the speed of gene evolution, it turned out that this whole genome duplication, which we found in sterlet, happened much earlier than the divergence of all sturgeon species. So it's more than 150 million years old, and it's common to all the, or to most of the sturgeon species that we could analyze so far. And now we cannot speak about whole genome duplications without mentioning Susumu Omuro, because he asked the question, what happens if a genome duplicates? We've just discussed it in the beginning. There is just twice the number of genes that you need as an organism to live. And of course, you can ask the question, what should happen with this additional set of genes? Of course, you can say that as you don't need them, they are just an additional burden, because it has to be replicated, it has to be packed into chromatin, gene regulation is a problem. So the first idea what would happen to a duplicated gene is that the best is to get rid of it. It's just degeneration, and then you lose it, and we call this a deduplication process. But Susumu Ono pointed out that there may be a big advantage, because you have now a lot of genes which you can use as a substrate for evolution of new functions, and this was called the neo-functionalization, so you can create genes with new functions, or you can sub-functionalize the function of the ancestral gene. It can be now taken by two genes, so you would have redundancy, you have new ways of regulating tissue-specific expression. So the question is what happened in the Sturgeon genome? And when we looked how many of the genes were still there in duplicate, it turned out that it's about 70% of these genes which are duplicated 150 million years ago are still there in 2 copies. When we compared several fish species and looked for this duplicate retention rate, it turned out that sterlet or sturgeons have the highest rate of gene duplicate retention, despite a very old whole genome duplication, much older than the events in the modern fish. And the secret why is, that's our interpretation, we found that the sterlet and all sturgeons have a very, very low evolutionary rate. So the rate with which genes are changing is in sterlet as low as in sharks or in the coelacanth. It's one of the slowest evolving organisms, which of course also tells us that it's very slow in adapting to new challenges by the environment, which of course is also a concern for conservation biology. The question, what are the mechanisms and drivers of deduplication? This is something that we really could answer and learn from the sturgeon. Some people say this mechanism of deduplication is just random, just by genetic drift. A gene gets lost, no problem, and then it goes over the whole genome. Others say, no, it has to be adaptation. There are processes where proteins are interacting in multi-protein complexes, genes are interacting, and of course it's important to keep the stoichiometry, and that this may be the driver of keeping either double the number or reducing it. So what does the sturgeon tell us? There was a first hint, you remember, I told you that several chromosomes which are listed here, and as you can see in the row where they have the protein and the gene number, some of them are really gene-rich, but they do not have a homologue. So meaning that a whole chromosome was lost early on. So this could give us a hint how this process happens. As we know, genes which are interacting in a function, they are not always sitting together on the chromosome. They usually interacting genes or protein are distributed over the whole genome. So this more points into a random process. And again, here where you see the red arrows, they point to these chromosomes of the sterlet where we have no homologue. Gray means there are only single genes on it. Red means these are regions which have an onologue. We call the duplicate from such a whole genome duplication an onologue. And what you see is that the genomic organization is in pieces. It's not just a continuous stochastic process over the whole genome, we see that there are islands of the genome where the duplicated region is obviously lost. And when we look in other fish which underwent recent whole genome duplication, it's the same picture. So this is in the goldfish. We also see this structured genome. This is the Atlantic salmon. It's the same picture. So what we can say is that this deduplication process is obviously segmental. Why is it segmental? Yeah? Of course, we still always would like to follow the adaptation hypothesis that genes that function together should be kept together in duplicate. So what we did is we made a comparison and looked at all fish genomes where we know that they underwent a genome duplication. Old ones like the zebrafish, but also the young ones like goldfish, and the sterlet is an old one. And what we found is that in all these genomes, there are only 27 genes which are commonly retained, and there are 191 which are commonly deduplicated. So this already speaks for a random process. And when we looked where these genes are, you don't have to read the table, but it turns out that these are mostly gene neighbors, genes which are together, which are retained, and also gene neighbors which are lost. So what we can say is that the deduplication is segmental. We know that it happens very early. So the massive deduplication is an immediate process after the genomic shock. The driving force for duplicate retention cannot be functional relationship as a main factor. There is a stochastic element to it. And a possible explanation, and this is more a wild hypothesis, but it needs to be studied, is that the conservation of the microsynteny, genes which are close together, is a so-called bystander relationship. We know from studies on gene regulation that very often a gene can have an enhancer or other regulatory elements which sit in the intron of the next gene upstream or downstream. And of course, this is a mechanism which really binds genes together structurally rather than functionally. So this was the basic research. But of course, nowadays we always have to ask, is there an applied aspect from the sterlet genome? And of course, there is a trivial answer. The genome is a blueprint of the organism, and it informs us about the physiological developments, the reaction norms, the pathology. If you are interested in an organism from any side, maybe conservation, biodiversity, but also economy, you need to know its genes, no questions. But especially with sturgeon, there are several, let's say, burning and also financial questions. As Vladimir already pointed out, sturgeons are endangered. The World Wildlife Fund has put the quote that sturgeons are the most endangered species group on this planet, at least when we talk about animals or vertebrates. Overfishing, habitat destructions affects the natural populations, diminishes the gene pool. So we need measures for conservation, and the genes tell us that. But of course, the big economic interest in sturgeon is the caviar. We look at the genome, and of course you find certain genes like the Zona pellucida genes, which in sturgeon have undergone a local gene duplication, so there are many, many copies. And these genes certainly are responsible for the eggshell, so they make the crispiness of the caviar. That could be interesting for the breeding industry. But the main problem is that, of course, only the females produce the caviar, meaning that about half of the sturgeons are not of value, namely all the males. And in sterlets, it takes about 4 to 5 years until a sterlet is half a meter. It has about 10 kilos. In the beluga sturgeons, they reach sexual maturity after 8 to 9 years when they are 2 meters and weigh 200 kilos. And so far, only by then, the aquaculture, the breeders were able to tell apart the males from the females. So it was quite interesting when we looked at the genome, and we found a region where, what you see here is a special analysis where the DNA of 30 males and 30 females was pooled, and then we sequenced these pools, and then we looked over the whole genome. How do these male and female specific reads align to the chromosome? And there is a very small region, and the region is so small it cannot be detected by conventional cytogenetics. That's why people never found sex chromosomes in the sturgeon. So there is a region here, and when you look for the blue, the reads, so the reads from the male, they are not mapping to this region in the female, so meaning the female has a piece of DNA which males do not have. And the same region in females, these are the red, is the red line, has only half the coverage of the rest of the genome. Meaning here we have a heterozygosity or a hemizygous region. This points to a WZ mechanism, which was already suspected but never proven. And now, where you see the red and the green arrows, we can make PCR primers. And these PCR primers are useful to determine the genetic sex, not only of the sterlet, but surprisingly for all the other sturgeons that we have tested, including the Siberian sturgeon. You get the product from the females because they have the W chromosome, and no product for the males. And this can be done with a small sturgeon, with a hatched larvae, with a fingerling, and then these animals do not need to be raised to old age or to high value. So this conservation of the sex marker is certainly important for the industry, but it also tells us that this WZ system in the sturgeons is old, older than 150 million years, one of the oldest sex chromosome systems, I guess, for fish at least, it's the oldest that we have seen so far. So this is the sturgeon story. It's going on with the genome of the Accipensa berry, which is an octaploid species. We are working on the genome. We have the first assembly. It's not perfect, so we have to do a little more to separate these 4 copies. The high C data look even more messy than what I've shown you. And I will just, with a few slides, point out to some other genomics projects which are in the lab. None of these is finished. It's all unfinished business, ongoing work. What we have done is to sequence the Australian lungfish. The facts about this is we have a very good genome, again, of chromosome size. The N50 is almost 2 gigabase. It's the biggest animal genome that has been assembled so far. We have assembled 37 gigabase of more than 40 gigabase genomes. So this is 15 times larger than the human genome. Over 90% of this genome are transposons and repeats. This fish is diploid. It has just a regular set of genomes. This genome expansion happened also very early in the history, and it's continuous and it's genome-wide. So these transposons have expanded over the whole genome in a more regular way. When we look into the transcriptomes, we see that these transposons are still active. So they are still jumping around. And the question is, why is this happening? Because we know a transposon can also jump into a functional gene and can kill it. But here, again, fish are maybe more resistant to something which is so detrimental for humans. The intron size, the average intron is about 50 kb in the lungfish. In humans it's 6 kb, but the introns make up 21% of the genome like in humans, so it tells us that everything happens more or less regularly over the whole genome. The structure has not changed much, so the arrangement of the genes and the chromosomal content of the lungfish is the same as we have calculated for the ancestral vertebrate karyotype. So in general, obviously, distance is not an issue, except for a very few cases, and this is one case from the Hox cluster. So Hox genes, they are determining the structure of the development of the vertebrate body plan, and down here is the mouse, and you see the whole Hox cluster, all the Hox genes are in one cluster, is less than 100 kilobases, and it has expanded in the lungfish and also in the axolotl almost 10 times, and those genes which are here, Hox1 and Hoxd3, they are separated in the lungfish by large distances, which obviously is no problem for the gene regulation. In the Hox cluster, genes are regulated together very, very nicely, except for this cluster from Hoxd8 to Hoxd11. This in the lungfish is also only 25 kb. So here is an exception for the rule, and it will be interesting to look closer to see what are the regulatory elements. Some genes in the lungfish are pre-adaptations for the conquest to land, so they have already genes which they needed to crawl on land, like olfaction genes for the detection of airborne odorants, surfactant genes for the lung, they are already there, and also the Hox genes which are important for making a limb. One thing that fascinated me, this is one thing that you learn as a student, that usually exon 1 is the largest, because it has the regulatory element. So the question was, what happens if these things expand? Is this just random? Is there something to it? And as you can see, still in the lungfish or in the axolotl, exon 1 is always the largest. So there is something to it which may be interesting to follow up. A final solution also was given for the question, who is the closest relative to land vertebrates? And it turns out that indeed it's a lungfish and not the coelacanth that was sold for many years as our closest relative. What is also maybe interesting is that besides the lungfish, the salamanders are also known for their super big genomes. And the analysis turned out that these are totally independent events. This happened twice. And it's other transposons which makes the genome of the lungfish grow than of the axolotl and the salamanders. What's next? Next is the genome of the South American lungfish, which we have started. And to our surprise, we are going to beat ourselves, because the South American lungfish genome is 90 gigabase, and this will be a challenge for the future. A small project that we are doing is this fish. So this is now an applied aquaculture problem. This fish is of commercial importance with a high market price, and it's also interesting because it's a sequential hermaphrodite. All of these fish are born as males, and at a certain age of 3 to 4 years, they transform into females, and then one can breed for the next generation. This is a problem for the aquaculture because this sex change does not occur in aquaculture. So there is no way to breed them. You have always to go out and collect females from the wild, which means that you cannot really do selective breeding and so on. So what Juliana, a postdoc in the lab, has done. She has treated undifferentiated juveniles with steroids and analyzed the transcriptomics, which only could be done after the genome was made, and it turned out that indeed it's possible to make females, so fish under estrogen treatment, they start to develop an ovary and express the genes which are of interest, so maybe we can produce these primary females for the aquaculture. This is also a genome that we have made some time ago. This is the arapaima. The arapaima is the biggest freshwater fish. It's very important for the aquaculture in South America. It's known as the Amazon cod. Its reach is also 3 meters in length, gets 200 kilograms, and also interesting from the biological side because it's a very ancient organism. And again, here's the same problem. One wants to know what will be a male or a female, especially for breeding. And when we did some analysis, again, from pool sequencing and rat tags, it turned out that this fish has an XXXY system, but by no means the sex chromosomes were visible in the karyotype. So when we looked at the genome, and we had a similar situation, as you see on this part, there is a region where there is nothing in the female genome and only half coverage in the male. And so we detected an XY system. This XY system is due to a genome, to a duplication of a little fragment. And when, we are going to confirm this, but again, we could develop a sex test for the arapaima, and this list tells you that it's a quite reliable system and it's much better than what they've been doing today when they do endoscopy or vitellogenin. The final few slides are about something which has no applied aspect, namely the question about why do so many organisms reproduce sexually when asexually reproduction is much simpler. The advantage is twofold. This is just rate of a sexual species when we have 4 offspring. You see the red line, that's a population growth, and this is the population growth for an asexual organism, because every individual is going to produce babies. There is no cost of finding a mate, but there is a disadvantage, because these asexual organisms, they don't have meiosis, which means they cannot get rid of deleterious mutations. That's at least what the evolutionary biologists can calculate, and it's one of the paradigms. There is a small fish from South America, which was the first unisexual vertebrate to be detected. It's all female. No males have been found. It's called the Amazon molly, not because of the Amazon River, but because of the Greek tribe of all female warriors, the Amazons. They had, of course, a trick. They were raping males from other tribes, and they were killing all the male babies. But the Amazon molly does it different. When we did the genome, we found out that the Amazon molly, the clonal female here, is a hybrid between 2 species. And this happened about 100,000 years ago. And there was a fish that is all female. And this fish does not undergo meiosis, but it needs a male, the sperm of a male, to activate the egg. The egg is diploid, and the male DNA is not included in the nucleus. And you can see, you can take different males, and you get always this all-female type. And but sometimes in the lab it happens, and this is that we got from a black father a spotted offspring, which was, again, pure clonal. And the mystery is that we find in these black Amazons these small microchromosomes, or B chromosomes, that were mentioned by Vladimir. So we were interested to know what is the biological meaning. are these microchromosomes bringing in new, fresh genetic material which helps to overcome the disadvantage of no meiosis. And we are doing now several approaches from genomics. And the most important thing is, and I have to load it, Vladimir again, when we found this, of course, the idea was we have to scratch out the microchromosome, sequence it, and then compare it to our other sequences. And this was the time I asked our human geneticists who can do it. And they said, oh, there is this Russian guy in Jena. He is a world expert. Talk to him. And this is how our collaboration started. We have now found these sequences, and we know that the microchromosome has about 84 genes. We have to do transcriptomics to see if they are expressed, and then this is something to do for the future. This is a team in Würzburg, the genomics team, and these are our collaborators in Texas and all the people from the Genome Consortia. Thank you for your attention.
Manfred, thank you very much. Maybe we have time for one or two questions, and I can start. Just your last slide, what I wanted to ask. So you mentioned regulatory elements, you mentioned transcriptomics, but for fish, it's like disaster, because the method we use for human in mammalians, for example, transcription starting site marks to find the regulatory elements. Yeah. But for fish, with multiplied, with polyploid genome, as well as in plants, it's like a disaster. So is it a good future for regulatory transcriptomics in fish? So how things are going?
I think, and this was also mentioned in the previous discussion, I think that's one of the most exciting questions in understanding gene regulations. Why are fish permissive to all these changes, and also amphibian, and why is it such a big problem in humans, and what can we learn for the humans? What have the higher vertebrates lost? And so one of the things that we are following is we always have the romantic idea from the textbook that there is one transcription factor that binds to one enhancer element, and then one gene makes 10 copies of the messenger RNA. What we have started in the Amazon molly, because it's a clonal fish, we are trying to see how big is the extent of stochastic expression variation. How tolerant is an organism to this, and then compare it, for instance, to human cell lines and see if we can manipulate expression levels and see what is the reaction. So there is a lot of future.
I see. Okay, so any questions or comments? Okay.
My English lives to be much desire, поэтому на русском, если позволите, спрошу. Содокладчик ваш подчеркнул, что особенности этих рыб стремление к полиплоидии, то есть кратное увеличение набора хромосом. Есть представители одного вида, насколько я понимаю, каспийский осетра и сибирский осетра. За период, я не знаю, миллионы или сотни тысяч лет, вот что произошло? Есть ли изменения на уровне кариотипа или же там какие-то другие изменения на уровне генома, там другого уровня? Спасибо.
Let me serve as a translator for you. So you just mentioned this genome duplication took place in a Sturgeon, one of the examples. But you mentioned those two species, was a kind of Caspian guy and the second is a Siberian Sturgeon. So just talking about these two species, how about difference among them? So evolutionary, so the multiplied genomes, have they changed if we compare to each other? Or is there a kind of difference in evolution of these two species?
I think for this, we have to wait until we have the genome. So in general, and this we also find if we compare very different looking fish, fish which are from the deep sea or come from the deep sea vents, and then a freshwater fish from the mountains, the repertoire of genes is not very different. So they all have the same gene. So it has something to do with genome regulation, and it's more a question of when and where and how many of the genes are expressed. And for this is a long way still to do.
Okay, thank you very much. Let's thank our speaker again. Oh. И, коллеги, у нас сейчас есть 10 минут, пока Даша у нас подключит. Мы предлагаем сфотографироваться на этом замечательном, на фоне замечательного баннера. И коллеги, организаторы нам, надеюсь, помогут, да? Нас фотографируют.